First of all, thank you, Garen, for the opportunity to present my work here in this seminar series. Thanks, Liam. It's been a super interesting talk. And today I'm going to be talking about partnering, time and space. And I'm going to start defining what partnering is. So we all know that all plants interact with soil microbes, but leguminous plants interact with rhizobial bacteria in a symbiotic relationship where rhizobia fix nitrogen for the plant in exchange for carbon compounds. There's a molecular dialogue that needs to be established between these two organisms, the plant and the bacteria. And the recognition through this molecular dialogue is the first step to the establishment of the interaction, but the outcome of the interaction depends on many factors. For example, the external environmental factors, such as the circadian clock, which will be the time side of this story, or the soil environment the plants are growing into, which will be the space. And then, there we go, yes. So from the paper we published in 2019, we observed two things. The first one is that the space, the status of the nodulation of the plant affects the way this plant interacts with the environment. The specific observation that we had was that the nodulation status was affecting the way the plant was moving nitrogen to the shoot. And then we wonder whether other things could be affected depending on the nodulation status of the plant. And in terms of time, we also observed that some specific nodule genes were regulated hours after nitrogen application. So we wonder whether it was just nitrogen or whether time was playing a role in the regulation of these genes. So let's talk about time first. And when we talk about time in plant science, we're talking about the circadian clock. And as you probably know, all plant metabolism, or mostly all plant metabolism, is regulated by the clock. And here you can see a simplified version of the molecular clock, the internal clock in Arabidopsis thaliana. And this is a series of transcription factors that regulate each other through feedback loops, and they're able to integrate signals of light and darkness photoperiods and also temperature regulation. In this internal clock, CCA1 and LHY play a central role, and where there are two genes in Arabidopsis thaliana, in the plant species, in the leguminous plant species that we study in the Gifford lab, Medicago truncatula, we can only find that there's only one of these genes. These transcription factors, they bind the sequence in the promoter of target genes that is called the evening element, and this is the sequence of the evening element. And in this way, by binding the sequence, they repress the expression of the genes, and it's called the evening element because it represses the expression of genes that are expressed usually in the evening. So because we thought there might be a link between nodulation and the circadian clock, we decided to get hold of two LHY Medicago mutants, one that has an insertion in the promoter region, another one that has an insertion at the end of the gene. And then we phenotype them for nodulation phenotypes. And what we can see is that despite not seeing any growth differences in terms of dry weight when we don't inoculate the plants with any rhizobia. So here you can see the wild type on the left and then the two mutants on the right. And there are no differences in plant growth when there's no rhizobia, and this is reflected by the dry weight. When we treat our plants with rhizobia, we actually see a decrease in plant dry weight in our two mutants, and this is reflected in this graph as well. And this decrease in plant dry weight is also reflected by a decrease in the number of nodules. So, okay, we believe there is a link between nodulation and the circadian clock, but we wanted to know a little bit deeper what is happening in those nodules. So we decided to perform a circadian clock experiment in our Medicago truncatula plants inoculated with rhizobia. So we grew our plants in a semi-sterile environment in perlite. We inoculated them with rhizobia, and we were growing them with a photo period of 12 hours light and 12 hours darkness. And then 6 weeks after inoculation, 5 or 6 weeks, we transferred them to continuous night. And then we sampled every 3 hours during the first 24-hour period and every 6 hours afterwards. And then we extracted RNA and we performed RNA-seq. So what did we observe? The first thing that we can see is that we observe oscillating genes with a period of 24 hours in nodules during in mature nodules. So there is a clock functioning in the nodule. And although you can see here in the heat map, we observed 2,832 genes, and although you can see it looks like it's a continuous wave of oscillation where the genes peak at maximum at different times of the day, we were able to perform cluster analysis roughly by dividing this data set into 4 clusters. So these clusters are represented here on the left. You can see the average gene expression in each one of the clusters and what kind of processes did we observe in each one of these clusters. We've got, for example, the dawn cluster that peaks at dawn here, where we find LHY. So we know this LHY is expressed in the morning, well, at dawn, and actually this makes all the sense in the world because it's repressing all the genes that most likely are going to be expressed in the evening. And the type of GO-terms that we see enriched in this cluster have to do with anion transport and isoflavonoid biosynthesis. We have another cluster, the morning cluster, where we see genes related to nodule light metabolism and cell wall biosynthesis. So it could be that here is where the nodules are starting to grow or recover from growth. We have the evening cluster where we find starch degradation, so carbon compound degradation. And we also have the late night cluster here in green, where we find GO-terms related to ureate biosynthesis and nitrogen transport. And since we find carbon degradation GO-terms in the evening cluster, and nitrogen transport related genes in the late night, we hypothesize that maybe nitrogen fixation is happening during the night in nodules. And then we wanted to know, well, what other things can we see in these clusters that are more related to the way the plant interacts with the rhizobia? How could be nodulation controlled at this level, and how is the clock playing a role? And when we looked into the other genes in more detail, we found that in cluster 3, that is the evening cluster, we actually find an enrichment in NCR peptides, which are nodule cysteine rich peptides. And we also find an over-representation of the evening element, which is the element that LHY binds to in all the genes in this cluster. So it looks like the clock could be controlling nodulation through NCRs and through their rhythmic expression. And we wanted to know whether we could find any proof that this was the case. So we performed a promoter analysis. This was work done by Minky Asham. And we performed a promoter analysis finding the nova motifs in the promoter of all the NCR genes in Medicao, which are more than 700 genes. And we can find, we were able to find the evening element in the promoters, and we actually can see that it's enriched in these genes when we compare it to the rest of the genome. So what are these NCR genes, you say? NCRs are going to be an important type of peptides that regulate the differentiation of bacteroids in the nodule, and they're going to be controlling the efficiency and the way the plant fixes nitrogen. So that makes them, they're present in most of the agriculturally cultivable legume species, and we believe they're really important potential targets to improve nodulation outcomes and increase plant production without affecting the above ground clock and the way the plant responds to the environment and light. So I hope I have convinced you that there is a link between nodulation and the clock. And now we can move on to space, and where the way we observed that the observation we got from the paper in 2019 was that the plant, the nodulation status of the plant was affecting the way the plant was uptaking nitrogen. So we wondered whether plants that fix nitrogen at different efficiencies, so that partner with different rhizobial species, whether they could be interacting in a different way with the soil environment. In this case, we wonder whether plants that fix nitrogen not so well and others that fix nitrogen much better, whether they recruit different microbes, and ultimately our idea is to find microbial cooperators that we could use to improve plant fitness. So I told you that different rhizobial species fix nitrogen with different efficiencies. And here you can see the way Medicatron catula grows without any nitrogen and without any rhizobia. So they're very sad. Here you can see how Medicatron catula grows when we inoculate it with Rhizobium meliloti 1021, which is not a very efficient rhizobial species for Medicatron catula, and this was proved in 2008 by Jason Terpelili. We can observe that when we inoculate with 1021, the nodules are cylindrical, they're pale, and generally the plants fix a bit of nitrogen, but they're not very happy. However, when we inoculate with high efficiency rhizobial strains, for example, Rhizobium medicae 419 and Rhizobium meliloti 1022, the plants grow a lot better, and the nodules are more pink, and there are more nodules, and the color of the nodules is indicating us that the nitrogen fixation efficiency is working a lot better. So we were wondering, well, how would these strains perform in natural soils, in agricultural soils? So we performed experiments with real soil. The way we did this was to germinate our Medicago seeds in sterile conditions. We transferred them to semi-sterile conditions. We treated them with the treatments that I talked about in the previous slide, and then we transferred them to soil. And we used 3 different soil types. I'm going to be using these colors all through the rest of the presentation, so it'd be good if you can remember them. We've got the yellow soil, the gray soil, and the blue soil. And in general terms, the yellow soil is the poorest soil of the 3. The gray soil is a medium nutrient available soil, and the blue soil is the one that contains more available nutrients. We grew our plants there for a while, and then we sampled when the plants were 66 days old. And how do our plants grow in these soil types? Here you can see a representative composite picture of how do they grow in the different soil types. So the first row is the yellow soil, the second row is the gray soil, and the third row is the blue soil, and you can see the different treatments and how do they affect. On the left is the picture, and on the right you can see the dry weight corresponding to these pictures. So if we focus only on the mock treatment, we can see what are the soil effects on plant growth. And here you can see that the yellow soil grown plants without any rhizobia are generally very sad. When we grow them on the gray soil, they grow a little bit better. And when we grow them in the blue soil, they grow even better. But what happens when we inoculate them with the different rhizobial species? So the results for the yellow and the gray soils are very similar. So we see there's an increase in dry weight when we inoculate the plants with the low efficiency rhizobial strains, but then when we inoculate them with the high efficiency rhizobial strains, we actually see an even bigger increase in dry weight. However, in the blue soil, we only see a significant increase in plant dry weight when we inoculate the plants with Rhizobium meliloti 1022. So in these experiments, we obviously took the pictures, we measured the dry weight, we also analyzed the mineral composition of the above ground, and we also analyzed the microbial communities in the endosphere. So I'll be talking about these results now. So in this slide, you can still see the picture of how our plants look like as a reference in the different soil types with the different treatments. And here you can see a heat map of the mineral analysis of the above ground material of these plants. So we basically analyzed by ICP-MS all these elements that you can see here on the right. And we see the results in the heat map separated by soil type and by treatment, and all the treatments are always in the same order as I've been presenting them up until now. So the way we did this analysis was we did the ICP-MS analysis, we took the results, we normalized them, we calculated the set scores, and then we used them to plot a heat map to see whether we could see trends of minerals that were accumulating in specific treatments and soil combinations. So one thing that I want to highlight first is to look at nitrogen, obviously, because we're talking about nitrogen fixation. And the trend that you can see here is that there is more nitrogen, the better the nitrogen fixation efficiency independently of the soil type. So we can even see significant differences between 1022 and mock in the blue soil. And then what else do we see? What are do we see clusters? Obviously we do. Here are the groups that are clustered here on the left, and I'm going to generally introduce what's in each one of these clusters. We've got cluster 1 that, as you can see, has higher values of in the blue soil in general, and it contains, for example, molybdenum, which is an important element for nitrogen fixation. We've got cluster 3 that is composed mostly of macronutrients, general macronutrients. We've got phosphate, sulfur, potassium, nitrogen, carbon, calcium in there. And we've got cluster 3, you can see it's got higher values in the blue soil samples, but also in the high efficiency rhizobial strain treated samples, both in the gray soil and somewhat also in the yellow soil. And we also have cluster 4, for example, that contains higher values only in the high efficiency rhizobial strains, only in the gray soil and the yellow soil. And it contains zinc, which is also an important micronutrient. And I told you we also did microbial community analysis. In fact, this is work that was done by Chrissy and Luke, and they're involved in the analysis. We analyzed the bacterial and fungal communities from all the bulk soil, the rhizosphere and the endosphere. But because I don't have enough time to present all the data, and I want you to stick with me for the rest of the presentation, I'm going to be talking about what happens in the endosphere, which is the root only for the bacterial communities. This is where we see the biggest differences as well, and the most interesting ones. So what happens in the endosphere microbial communities? The first thing that I have to tell you is this is a PCA plot. It's basically like a PCA plot where it shows the bacterial community structures on the different soil types and the different treatments. And what we can see is that the microbes that are recruited inside the root, the structure of the communities mostly depends on the soil type, because we can see that the yellow soil samples cluster here, the gray soil samples cluster here, and the blue soil samples cluster here. However, are there differences due to the high nitrogen fixation efficiency strain treatments? When we look into this into a bit more detail, you see I've circled the high efficiency strain treatments with a black circle, and we can see there's a shift in the community structure compared to those in the mock and 1021 treatments. We can observe this in the yellow soil, in the gray soil, and to a lesser extent, but also it's there in the blue soil. Okay, so we've done the mineral analysis of the above ground plant material, and we know that what are the community structure in the endosphere, what is the community structure in the endosphere of all these plants. So what we did then was to try and correlate these two things and find out whether there was any correlation, because obviously microbes that are recruited in the root might affect the way the plant moves elements to the shoot as well. So you can see the results in this plot. This is a CCA plot with 2 axes. It's a 2-dimensional one. So you can see the microbial community structures here at the bottom with the same points that were in the previous slide. And then you can also see on top the arrows with each cluster, and with an asterisk, you can find the ones that actually correlate significantly with part of the data. So here we can see cluster 1, for example, that we said that had higher values in the blue soil, and it's actually pointing towards the blue soil samples. So it's defining the way these communities are structured in comparison to the other samples. And for clusters 3, 4, and in this case also 5, what we can see is that they point down, and they separate the high efficiency rhizobial treatments in both the yellow soil and the gray soil when compared to this. So that means that there's a correlation between the microbes that are being recruited in the root in these treatments and the mineral composition of the above ground. So what are actually, do we have specific examples of these microbes? And I don't have time again to go through all of them, but I'm going to give you two very important examples that we found. Because sometimes when we treat with the rhizobial, high efficiency rhizobial species, there's recruitment of more of some microbes and less of some others. So I'll give you one example of each. Here is the example of Allioflea, for example, that is from the order of Rhizobiales. We cannot detect any Allioflea in the soils, but we see there's more accumulation of these bacterial species, the better the nitrogen fixation is. And obviously, because it's part of a Rhizobiales order, we believe it might be hijacking the system, just getting inside the root, because it looks like our Rhizobial inoculums. And then we have the opposite case as well with Otaequangia from the order Cytophagales, that it's more abundant in the gray soil, in the bulk soil. However, it gets more into the root of those plants that haven't been inoculated with any rhizobia, or that are, the inoculation is low efficiency. So it could be that these microbes are opportunistic and they're getting inside these plants because they actually are not the ones that are performing the best or mobilizing the best nutrients, but it could also be that the plants are recruiting them to cope with these nutrient stressful conditions. And we really don't know, but we are going to perform these experiments in the future with a PhD student that has just joined the lab, and we hope we can understand a little bit better about these bugs. And I'm just going to finish thanking everyone that has been part of either the time or the space project, and yeah, I'll take any questions.
Okay, thanks very much, Beatriz. Wow, that's an absolutely giant data set you've got there of minerals and bacteria and fungi, which you didn't show or anything like that. So wow, that's very impressive. So you know, I'd encourage everyone, people need to gather their thoughts before they ask a question, I think. So let me kick off by going back to the first example when you were talking about the clock. So you were looking at the clock in the nodules. So do you think the clock that you see in the nodule is going to be the same as the clock that you'd see in the leaves or other tissues as well?
Well, it seems to be regulated by LH1. It seems that it's playing an important role. So I would say they're probably synchronized very well. And we do see, we've got also leaf and root samples from this data set, and we've performed qPCR and we can see them oscillating equally. Just we haven't done RNA-seq analysis on those other samples. So, but that would be an interesting thing to do, just another enormous data set to analyze as well.
Why not? That's true. So I've got a couple, as people are getting their thoughts together, I got a couple of more technical questions, I guess. So the LHY mutants that you looked at, so those are insertional mutants. Do you get those from the Samuel Noble, or do you get them from somewhere else? Where do you get those metacardial
From
mutants?
the Noble.
Okay.
Yeah.
Okay. So you just order them and they come for you, and that is a resource which is available for people wanting to do reverse
Exactly.
Okay. And then secondly, about the second,
brilliant.
Yeah, the experiment about the endosphere. So is that the case of you literally dig up the soil and then just sequence the bacteria which is in the soil, basically?
Well, in this case, yes, we didn't do whole genome sequencing. So we amplified the region of the 16S gene, or the ATS for fungi. But yeah, we did that for the bulk soil, and for the rhizosphere and for the endosphere is just washing the roots, making sure you only get the ones that are inside.
Yeah, I see. Okay, so a couple of questions. So first one from Gaurav Pandiharkar, sorry about the pronunciation. So have you performed nitrogen fixation assay for different rhizobacterial strains? And if yes, do they show similar results to your dry weight data?
We haven't done nitrogen fixation assays. No, we haven't done the ethylene assay. No.
Okay.
We just look at the way the plants grow, and they've got the same phenotype independently of the system that we really grow them into, because even in the soil types you can see the differences. I think it is important to see that even in the blue soil we can see an increase that contains lots of nutrients. We can still see an increase in dry weight when we treat with one of the high efficiency rhizobial strains.
Okay, so a question from Stuart Sullivan. He asks, do you see period difference between the shoot and nodule samples when you grow them in constant light?
We cannot say we see it. Let's say no. That doesn't mean it doesn't exist, but we can say we don't see it. No, that would be simple.
Okay, so another question about the clock here from Dae-Kwan Ko. So in other plant species, LHY is functionally redundant with CCA1. In Arabidopsis, LHY single mutant has a slightly shorter period than 24 hours. Have you checked the clock function is disrupted in the LHY mutant in Medicago?
Yes, we have, and actually we're doing even more reps in different conditions at the moment, but we can say that it is also, we've measured the leaf movement. I just didn't have time to put all the videos and so on, but it seems to be shorter in Medicago as well. So it's the same case as in Arabidopsis.
Okay, I've got I guess a follow-up just for my own curiosity. So when is it that Arabidopsis? So when is it that you get the CCA and the LHY together, and what's the split of plants that have just one, or the other two?
So Medicago only has one of the genes. That it only has one, and the Arabidopsis has two. They're functionally redundant, as he's saying as well.
Okay, and okay, that's fine. Yeah. Okay, so question from Catherine Evans, and she asks, why do you think the type of rhizobia can affect the concentration of nutrients other than the nitrogen?
Well, in this case, I believe it could be due to the other, or the way the plant responds to rhizobia. It could be a defense response, depending on how efficient the nitrogen fixation is, and that might disrupt other pathways. But it could also be the way these plants are recruiting other microbes, because these microbes, obviously, we've talked about what happens in the endosphere, but it could be that we see few changes in the rhizosphere as well. So obviously, it could be that they are recruiting. They recruit different microbes, and these microbes might be helping or making some minerals more available for the plant or less available.
Yeah, indeed. You're changing the environment there. So a question from Sarah Lopez Gomalon. Do you think that the different ions available in the soil may affect the release of molecules from the plants that can prime the interaction with the bacteria? This happens in fungi.
Yeah, definitely. So if we compare the mock treated samples across only the mock treated samples, we can see differences, because obviously the root exudates are going to be different depending on the soil type. I would expect that, yes, and there would be, but we're more interested into knowing what happens across one soil type with the different treatments. But yeah, obviously that would be a great I would expect even bigger differences in that case.
Okay, so a question from Ben Gu. Did you try different original soils to test the rhizobial fitness? Is SM1021 worse than A17 in all the soils?
So yes, it is, as I showed on the I can go back there if that's better. We can definitely see that 1021 doesn't work very well, and not as good as the high efficiency ones, at least on the yellow and the gray soils. And yes, we tried this. These are 3 different soil types, so definitely.
Okay, yeah. Okay, and a question from Sonali Roy. Do you see any difference in carbon metabolism genes or transport genes in your circadian rhythm study of nodules?
I cannot tell you right now. I think we do, but I don't remember. We've also done RNA-seq analysis with the different rhizobial strains, but you're talking about the circadian rhythm. So in that case, this one I don't remember.
Okay, it's unfair to ask you to remember every single gene in those giant data sets. So I think that's the last question. So I'm going to thank you, Beatriz, for giving your talk. It sounds like that work